Hi, Michelle Glass here, and our topic for our Chapter 3, Lecture 3 series is what I like to describe as organic chemistry. Organic chemistry is important for us because these are the molecules that make up our cells. So not only are these the cells that our bodies need and other cells need, in order to make the cell, but then also do all the cellular functions. But these then are also the molecules that we're eating, because when we're eating food, remember, we are eating other organisms, both plants and animals, and so we are eating the cells and we're eating those molecules. So these are the building blocks of our cells. These are the things that we're eating for food, where our bodies are breaking these molecules down in order to make the molecules that our bodies need. And we'll see that that is true as we progress through all the different types of cells that we'll be talking about in the future. By definition, organic macromolecules are carbon and hydrogen-based molecules. And the significant atom here is really that carbon. If we look at our periodic table of elements, we see that carbon has atomic number of 6. And so we can draw out our carbon atoms, and we're going to put, of course, 6 protons in the nucleus, 6 neutrons in the nucleus, and then 6 electrons orbiting the nucleus. And when we're looking at the valence electron shell, we will see that there are just 4 electrons, 4 valence electrons. And so that tells us then that carbon can form actually 4 bonds. And another way to represent this is by using your carbon here, your C for your chemical symbol. And then, of course, these are our covalent bonds. They're representing a pair of shared electrons. And so we can see here that carbon is able to form 4 of these. So this is just sort of a shorter way of representing this and going through the whole process of drawing out the the atomic structure for carbon. Now, when we talk about carbon as a carbon and hydrogen-based molecules, what we see oftentimes is these extra bonds are filled up with hydrogen. So that's what we'll be looking at. Now, for me, whenever I think about organic molecules and I think about the job of carbon, it reminds me of a toy that I played with as a kid. This is kind of an old toy now, so you may or may not be familiar. The Tinker toy is the toy that I'm thinking about. And in the Tinker toy, it was like a construction toy. And it was all made of wood. And you had these circular pieces that had holes in them. And then you had the sticks. And you would poke your sticks into the holes of the circle piece. And then that circle piece was that major connector piece that allowed you to build all your different constructions. And so I automatically just think about carbon as that circle piece in the Tinker Toy construction set. So when we talk about building our organic molecules using carbon as that connector piece, we'll be able to see that carbon, because it can make the 4 bonds, can form these really big molecules. And we call really big molecules macromolecules, where, of course, the prefix macro means big. All right, as we look at our different types of macromolecules, just like you can make all sorts of things with those Tinker toys, you can make all sorts of things using that carbon. And so we will see our macromolecules vary in 4 important ways. They can vary in length. Now, all I've done here is just my shorthand. So I've shown my carbons connected, and then I've left these other bonds kind of empty, because really it could be another carbon, it could be hydrogen, it could be oxygen. Typically, when they're left empty like that, the assumption is that hydrogen is going to be filling in the bond there. So we can see that they vary in length. They can be super duper long. They can have extensive branching. They can have double bonds. Remember, a double covalent bond means you're sharing 2 pair of electrons instead of 1. And they can also form rings. And very often you'll see your ring structure is abbreviated in this way, where each one of those points is representing a carbon. And then, of course, to complete carbon's outer shell, you would have to have bonds with other atoms, usually hydrogen, oxygen, maybe other carbons. So these are just sort of our shorthands to see different ways that we are going to get variation in our macromolecules, and how that center circular piece of the carbon can be really useful in constructing a wide variety of molecules. As we look at our macromolecules, we'll see that each of them will have what we can describe as a building block, and our fancy term for this is a monomer. So mono means one. So monomer is like the basic building block. If you were thinking about maybe Legos here, then each Lego brick would be a monomer, and as you put those Lego bricks together for your construction, you're building, in the case of our chemistry, you're building what's called a polymer. So that prefix poly, of course, means many. So a polymer is when you have many monomers connected together. So in order to get a macromolecule, a really big molecule, you need to have monomers linked together. And so that's what we're going to be looking at as we progress through. Now, the thing that's interesting to note is that our monomers are universal. So we'll see these in common in all cell types, whether you're in domain archaea, bacteria, or eukarya, whether you're in kingdom plantae or fungi or animalia, you'll see those same sets of monomers. The thing that is different, the thing that is special, is that each cell then, each type of cell, each organism can combine those monomers together in different ways to make different polymers. So polymers can be unique to the organisms or cells, but they're all using the same monomer. So this is another kind of universal aspect that we'll see with living things. In order to connect those monomers together, in order to connect those Lego bricks, you're going to do some chemical reactions. So we've already, I think, introduced this idea of a dehydration synthesis reaction. So the term synthesis means to make, and dehydration, you already know that word, that means to pull out water. So what I've done here is I've used some abbreviations. This R is standing for any type of monomer. So this could be any combination of carbon and hydrogen and maybe oxygen that would be there. Remember, it could be really small, it could be really big and complex. We looked at variations in length and branching and double bonds. So that R there is kind of like in algebra when you use the X to represent any number. The R here is just going to be a generic kind of abbreviation. Now notice in my monomers I have both a OH, a hydroxyl group, and a hydrogen group. So when I do my dehydration synthesis, what I'm doing is I'm going to pull out OH and H. If I pull out two hydrogens and an oxygen, I've removed water. So that's my dehydration part. But I still need to complete these bonds, and I do that by making a new bond between those monomers, and I'm building here my polymer. So dehydration synthesis reactions are where you're going to pull out a hydroxyl group, an OH and an H, to form water. And in doing so, then you're going to create a new bond between your monomers in order to build your polymer. Now remember, I said that all cells are able to take those monomers to build these specialized polymers that they need. And then I also mentioned that because, we're eating cells when we eat food, and this is true for all organisms, if you're consumers or decomposers, you're going to then take those polymers and then break them down so you can then use those components, those monomers, to make the polymer specific to your needs. So this is looking at how this cell can make the polymer. If you're eating food, you're going to need to break that polymer down. And so we can see this, and what is the opposite? This is our hydrolysis reactions. Remember, hydro here is standing for water, and lysis means to break. So literally, we can think of hydrolysis as breaking our water molecule in order then to break down our polymers. So this is an example, remember, of a catabolic reaction or a breakdown reaction. And this is exactly the opposite of what we just looked at. So we're going to start with that same polymer. We're going to take that water molecule and break it apart into a hydroxyl group, an OH, and a hydrogen. And that's going to allow us then to break our polymer down into monomers. So when we eat food, we're going to do hydrolysis reactions in order to get our pieces and parts. And then we're going to use those pieces and parts, those monomers, in order to do dehydration synthesis reactions in order to make the polymers that our cells need. And this is going to be, again, universal thing that's happening with all cell types, no matter what domain that you're in. Remember when we looked at our properties of water, one of the important properties is that it's reactive. And so here we're seeing that to be the case. Okay, so we've already sort of started talking about this, although you didn't really realize it. We will see what are described as functional groups in our organic chemistry. So these are groups with the same collection of atoms, and they have like a distinct set of properties. So we do need to be able to identify these different functional groups. So this is pretty much just a memorization practice. Now we're using our letter R here again as our generic abbreviation for any other kind of component here. The key thing to look for first in our amino group is the nitrogen and the 2 hydrogens. Now this we will see functioning like a weak base. We also have what's called the carboxyl group. Carboxyl groups have a carbon paired with an OH. We're going to call this a hydroxyl group. We've already used that term. And then double bond O. We'll see this functioning as a weak acid. Oh, and I kind of messed up in my presentation, so take a look right here. I know I've squished it in and made it really tiny. This is our phosphate group, so let's make sure we're writing this down in our notes. Phosphate group, look, is a phosphate, and then it's going to have 4 oxygens around it, one of which is a double bond oxygen. And then notice that these oxygen atoms all have a negative charge. That's going to be important as we talk about this phosphate group in our nucleic acid molecules. One of the important features of the phosphate group is that it can store energy. So we'll see that in our energy molecule as we go forward. Here then is that hydroxyl group that we've mentioned previously. We saw this in our discussion of dehydration synthesis and hydrolysis reactions. We saw it also as an important component to the carboxyl group. The hydroxyl group is the OH, and we'll see them in alcohols. Here we have what's called the carbonyl group. So the carbonyl group is carbon double bond oxygen, and then here we could put R's here to say that we're finishing up this bond. Maybe here you have an R group and here you have a hydrogen. So of course there's variation that can exist there. And we should have one more, which is our methyl group, CH3. Now we'll see methyl groups can be added on to genes or taken off of genes, and in this way it can affect or to some extent control gene expression, which we'll be talking about more this semester. Okay, so again, you need to be able to list out your functional groups and also identify the atoms that are involved there. So I would just practice kind of writing those out and drawing out those different functional groups. I've also, of course, got these embedded in your Quizlet sets if you want to use that for study practice. You know, as we are talking about our organic macromolecules, or we can talk about them as biomolecules, meaning molecules of life, there are going to be 4 categories, and we're going to have video presentations on each of these. So we're going to be investigating further the carbohydrate, the lipid, the protein, and the nucleic acid. As we look at each of these, you are going to want to pay attention to, you know, what atoms are involved, what are the functional groups that you find here, what is the monomer for each, and then we'll be looking at things like specific examples and functions of these molecules in our cells. So stay tuned.